Hello, everyone, and welcome to today's broadcast, Whole Genome Identification of CRISPR Targets, presented by Dr. Dimitri Perrin, a senior lecturer and science and engineering faculty in electrical engineering, computer science, and data science at Queensland University of Technology. I'm Dr. Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on that Ask a Question box on the far left of your screen. Type the questions into the drop-down box that appear on the screen, and our speaker will respond to your questions via email. If you have any trouble seeing or hearing this presentation, just click on that Ask a Question box and let us know you're having some trouble. This presentation is educational and thus offers education credits. Please click on the Continuing Education Credit tab at the top right corner of your presentation window and follow the process to obtain your credits. Without further ado, please join me now in welcoming our presenter, Dr. Dimitri Perrin. I will now turn the presentation over to him. Dr. Perrin, welcome.
Hi, well, hello, everyone, and thank you for joining. Thank you for the introduction. Today I'm going to talk to you about more of a computational side of looking at CRISPR, and I'm going to talk about how we can design algorithms to identify good CRISPR targets in an entire genome. So the way the talk is structured is that I will start by giving you a brief background and motivation as to why we want to identify these targets in entire genomes. Then I will explain in the specific context of knockouts what kind of targeting strategy we developed. Then we'll explain how our algorithm is filtering targets so that we only select what we think are going to be good targets. I will show you some results in the sleep context, and then I will explain the types of challenges we're currently trying to solve. So just to give you some initial background, I'm currently based at Queensland University of Technology in Australia, but I started working with CRISPR when I was in Japan in a group that is focused on circadian biology and sleep. And in that field, like in many other fields, researchers rely heavily on knockouts, especially using mouse or fruit flies as model organisms to study specific genes. So for instance, there's a famous paper from 2006 looking at a BMOL1 knockout to study its importance for the circadian clock. Similarly, this paper from 2011 on CRY1. And so because of that reliance on knockouts, for a long time, the progress was slowed down by that mouse knockout generation, because it can be a relatively long process. For each generation in your knockout process, it takes roughly 3 months, and you have multiple crossing to obtain the allelic knockout that you're looking for. So in that context, any method that can help accelerate that knockout generation process is likely to have a very significant and very positive impact. And CRISPR was just the latest in a long list of tools people have used, zinc fingers, talents, and so on. The main advantage with CRISPR in theory was the ease of use and the potential efficiency of the method. That's why people got very excited about that technique. That's how we started working in that space. And on paper, is very nice, but what we found back when we started about 5 years ago was that in the first generation, the mice that we were looking at contained a mosaic of wild type and knockout cells. And the rate of complete biallelic knockout was relatively low. It was around 60 to 80% at best. And also it's even though if we have a good CRISPR target, the results are promising, it's actually not that easy to design a good target. And so based on all that, our objective was to develop a method that we can detect targets that will be efficient and safe. So we'll define these two terms in just a moment, but use that sort of detection strategy to develop an algorithm that will detect targets in the entire genome. That one of the objectives in doing that was to increase the reproducibility of the work, and also to make all the targets that we identify publicly available. So introduce two properties, efficiency and safety. So we'll give you more details about what I mean exactly. So in terms of efficiency, well, if you have a low knockout efficiency, in a way, you're only getting half of the promised benefits of CRISPR. Of course, you still have the ease of use, which is nice. It's nice to have a technique that's relatively easy to use. But what we also want to eliminate, if we want to accelerate the development of these new knockout lines, is the need for crossing over multiple generations, because that's what's really taking a lot of time. So what we want to achieve is highly efficient production of whole body complete biallelic knockout in a single generation. And what we mean by highly efficient is that we would like at least 90% of the offspring to be a complete knockout. To achieve that, we need to rely on the selection of the targets. So we need to be careful about which targets we're going to use. But in the knockout context, we can also play with the actual targeting strategy. And so we'll cover these two aspects during the presentation. The other aspect is the safety of the targets. Of course, we want to knock out a gene. So we want to be efficient in knocking out that gene, but we also want to make sure that the guides that we generate are not going to make a modification somewhere else in the genome. Because otherwise, well, if we observe an interesting phenotype, we have no way to know if that phenotype is a result of the gene that we're knocking out, or if it's just a side effect of an unexpected modification somewhere else in the genome. So that means first that we need to exclude targets that occur in multiple locations in the genome, because we have no way to know where it's going to cut. But we also want to eliminate targets that have partial matches in multiple places in the genome, because it has been shown that the guide RNA doesn't need to have a perfect match to the sequence in order to bind. So we need to take these two aspects into account. Our perspective is that we have to be very strict about this, even if it means excluding many potential targets. That's especially going to be true in terms of possible long-term application of CRISPR methods. In the context of what the group is doing in second biology, well, it's bad if we have enough targets modification that leads to us being misled when we're studying a specific phenotype. But I mean, in a way, it's just going to delay our own research. But in some applications, in health or in crop modifications, well, the impacts would be much larger if we have unpredicted off-target modification. So that's why we took an approach to be very strict about eliminating targets that we felt had a risk of off-target modifications. Of course, when I say we need to ensure that guides don't lead to off-target modifications, well, it's difficult to really guarantee this, but what we try to do is minimize the risk as much as possible. So that's what I meant when I said we want targets that are going to be efficient and safe. The other thing I mentioned is that in the context of a knockout, we can play with the design of the targets, but also with the targeting strategy itself. What I mean by that is that there's nothing stopping us from targeting multiple sites in the same gene that we trying to knock out. And so we developed a nice little model that tried to explain why that could be a good idea. And so the idea is that, of course, you have multiple processes that are involved when you're using CRISPR. But if you have a very simple description of what's happening, you can describe the efficiency of your knockout as the ratio between your concentration of your guide RNA and the dissociation constant plus that concentration. That's if you use one target. But if you use multiple guide RNAs for the same gene, well, what you have is that the probability that you're cutting is going to be 1 minus the probability that you don't make a cut anywhere else. So for instance, if you have three, if you're targeting 3 positions in the same genome, you get that equation at the bottom. Of course, cutting is only half of the of what's happening. The other side is that there is a recovery rate. There are repair mechanisms, and so it might be that you're making a cut, but that the repair from your cut is actually not leading to any knockouts. And so we measure that. We take that into account with our parameter alpha, which we describe it as the recovery rate from that cut. And if you that the probability of having a cut is going to be that ratio we had before, S over K plus S, multiplied by 1 minus alpha. That's for just one side. And as before, we can extend that. So if we have 3 targets, we get the formula at the bottom, but we can generalize it to any number of targets, and we get that complex looking equation at the bottom, which might look slightly scary at first, but the interesting thing is that, well, all these parameters, we can either measure them or just estimate them and just have simulations of what the expected efficiency will be. And so the interesting thing is that we get results like this, where for a fixed guide RNA concentration, the more guides you use, the more efficient the knockout is likely to be. Of course, you have diminishing returns. So if you use 2 guides instead of 1, you get a big jump in the efficiency. If you use 3 instead of 2, it gets better again. But then at some point, if you use 25 guides instead of 24, well, there's not that much of a difference. And so the idea is, well, to have some balance, we need something practical. So the value we settled on was to use 3 targets, so to use triple targeting. So that's what we tried. First for the tyrosinase gene, which is a gene that's responsible for the black color of the fur of the mice. And so we designed 3 different targets for that gene, and so tier 1, 2, and 3. And so you can see at the top of the image, for each target used individually, the mice that are completely white, the efficiency is between 36 to 65%, which is in line with what we expected at the time. But the interesting thing is, if you deliver all targets at the same time into the same embryo, then all but one of the pups are completely white. And so we had 40 pups in the offspring in total. So that means a 97.5% first generation double knockout, biallelic knockout efficiency. So we were very excited about that, but in a way it's a blessing and a curse, because the blessing side is that, well, it looks like it's going to be very highly efficient. We can generate knockouts in the first generation very efficiently, so that's very exciting. But there are 2 limitations. Well, the first one, the main one, is that for each gene that we want to target using that strategy, well, instead of using 1 target, now we use 3 targets. So if it was tricky to design 1 target, it's going to be even trickier to design 3 good targets. The other thing that we have to remember is that, well, we want targets that will minimize the risk of targets, of target modification. And of course, the more targets we use, the more likely it is that there is going to be an off-target risk. If we use 3 targets, we multiply the risk of off-target modifications as well. And so that means that this strategy of triple targeting for knockout only becomes a practical solution if we have a way to quickly detect, quickly identify good targets, and ideally save them once we've detected them for the whole genome, so that anytime we want to target a new gene, we very quickly have access to good targets. And so that's where we got the motivation to design an automated method to do that. So the first questions we asked ourselves is, well, what does a CRISPR target look like anyway? And so the basic rule that I got from my experimental colleagues was that, well, they were looking for a sequence that had a fixed length, started with a G and ended with GG, and that for knockout, they didn't really care on which strand that target was located. In practice, they were sort of a little bit flexible about what they were looking for. They told me, well, you can start with A or C, it's fine. But what's really important is that, as I said before, that sequence is going to be unique. And they also told me, well, the secondary structure is going to be important, and I'll get back to that in a few minutes. And also, we want to be careful about the off-target effects, as I mentioned multiple times already. And so that sort of gave me the initial skeleton for what we wanted to do. And so based on these initial guidelines, I started to develop a detection method. So the objective is to have a method that will take as an input a genome and will detect all the targets for all the genes in that genome. So it's a slightly different approach to some tools that basically do an on-the-fly detection where you provide a sequence and it's going to find targets just for that sequence. The reason why we used whole genome detection is that, well, in the lab, there were different people interested in different genes. And then we realized that, well, many different groups will be interested in that kind of resource. They would all have different interests. So it just made sense to just process the genome all at once and just save the results. It's more efficient. In terms of the technical details, the method is implemented in Python and in C. And it's using also some third-party tools like Boteye, for instance, for alignments. The way it's set up, we can detect targets for a specific gene if we really want to just work on a gene or a list of genes or the whole genome. In practice, because of memory limitations, the way the whole genome targets detection works is that we take the list of genes for the genome, we create a list of 20 to 25 genes, and then we process the one list at a time, just because otherwise we have some memory issues when we run the method. And so in the next few slides, I'm going to detail how that detection method works for one gene. It would be the same. I mean, basically, once we're detecting for the whole genome, it's the same process. We just do that for every single gene. And so I'm going to assume that we detect targets for just one gene, and I'll detail that, exactly what the method is doing for that hypothetical gene. So in the first step, what we're doing is that we want to extract all the possible targets for that gene that we're targeting. So what we do is that for all suitable exons, we extract all the targets that match the basic pattern that my colleagues were looking for, which is something that starts with A, C, or G, then has 20 any nucleotide, and then ends with GG. Because we don't care about the strand, we also look at targets that, at sequences that match the reverse complement of that pattern, because it's we don't care about the strand. And in terms of what I meant by suitable exon, is that here we want to knock out a gene. So that means we're only going to consider exons that are shared between all the isoforms of that gene. And that's because we want to just knock out all the different versions of that gene. Of course, for different applications, that rule might change, but in case of knocking out a gene, that's a very important criteria we have to take into account. Once we've done that, once we've extracted the sequences that match that pattern, we know that all the potential CRISPR targets for that gene are in the list. So the only thing we have to do now, and of course it's a very important thing, is to go through that list and remove what we consider might be bad targets. And they're going to be bad either because then we think they're not going to be efficient, or because we think they're not going to be safe. Okay, so step one, extract all the candidates, and then all the remaining steps are about filtering that list, so that we're at the end we're only left with what we think are going to be safe and efficient targets. The first step is to remove multiple matches, because as I said before, we want to knock out the gene we're interested in, but we don't want to modify anything else. So we need to have a very high degree of confidence on where we're going to make the cut. So if we have a guide, a candidate sequence that we detected more than once when we extracted the sequence, we reject it, because we already know it's not unique. Then what we do is that for all the sequences that passed that first test, we align each of them against the entire genome using bot2, and if a candidate has more than one exact match, we reject the target. Because if we use that target, well, we're going to have a cut in the gene that we like, but also somewhere else in the genome, and so it's not a suitable target. Step 3, we're looking at the GC content. So at the time, when we started the project, there were different guidelines about what would be a good GC content for the target. Many of them were conflicting, and so at the time we just opted for a GC content of at least 55%, and also all the targets that failed to meet this criterion are rejected. In practice, it's probably better to avoid extremes. So at the moment, we're just running different tests of the method with different ranges. So for instance, the GC content between 25 and 75%. In practice, when we look at the results, it looks like targets that would have an extreme GC content, even if based on the criterion we use here, they pass that step, they would usually fail somewhere else, either because they end up having partial matches somewhere else, or because they have a very poor secondary structures, or things like that. So it looks like that step, even though it's filtering many targets, in practice, if we remove it, these rejected targets would be rejected somewhere else for a different reason. We might, it looks like the exact value here is not super important. In step 4, we remove the targets that contain that polythymine subsequence, because it's transcription terminator sequence, and so my colleagues wanted to eliminate them. The advantage is, as for the GC content, it's something that's very easy to detect, and so it's a step that is going to very quickly eliminate targets. And because overall the method is quite computationally expensive, it's nice to have some steps that are actually quite fast, because the more targets we reject early on, the less things we have to do with them later on. So it's always worth removing things quickly where we can. In step 5, we look at we take into account the primers that were used in the experiments that we were doing. And so here I give you the sequence of the reverse primer that my colleagues were using. And so they wanted to avoid targets that were too close to that primer. So what we did is that for every sequence that made it that far, we calculate the similarity between that sequence and the reverse primer. And if they're too similar, we just reject the target. Of course, not everyone is going to use that primer when they're building their guides. And so we have the ability to just switch off that step if it's no longer relevant. In step 6, we look at the secondary structure And the reason why we do that is that the secondary structure of the guide RNA is very important for the efficiency of the target. The 23 base pair, including the PAM motif that you generate, well, is important to bind to the region you're targeting, but the rest of the guide is important to bind to Cas9 and bring Cas9 to make the cut. And so what we do is that we generate the entire sequence for the guide RNA, so including the targets and the rest of the guide. We calculate the secondary structure of that entire sequence, and then if the part of the guide that's used to bind to Cas9 has a poor structure, and if that structure is stable enough based on the energy, then we discard that target because we say that, well, it's not going to be efficient because it won't be able to bind correctly to Cas9. So once we've cleared step 6, we're only left with targets that we think are going to be efficient. The other thing we need to check, of course, as I mentioned at the beginning, is how safe they're going to be. So we took that into account already to some extent in step 2 when we removed the multiple matches. But what we know is that there are other sites in the genome where the guide might still bind because it has only a partial mismatch. And so there are different strategies to look at that. Some people just count the number of sites that have one mismatch compared to your guide, number of sites that have 2 mismatches, 3 mismatches, and so on. But there was an interesting paper from Feng Zhang group in Nature Biotechnology in 2013 that looked at the specificity of the binding, and they found that that specificity doesn't depend just on the number of the mismatches. Also, the position of that mismatch is important. The way they did that is that they took one sequence and then generated multiple sequences with just one mismatch at different positions, or 2 mismatches at different positions, 3, and so on. And they had, let's say, the binding efficiency. And based on these experimental results, what they came up with is a rule and a scoring system. So the rule is that they say only need to worry about 4 mismatches. So for each target, we find them. And then they have a scoring system based on the number of mismatches and where that mismatch is located. And so you can see W, at the bottom you have the exact scores. So the position of the mismatch is going to have an influence on the score. So for instance, the first value in W is 0, which means that, well, that's a mismatch here is going to have a big impact, whereas if you look close to the end of W, you have 0.804. Well, because we take 1 minus W, well, is closer to 0, and so the impact of that mismatch in terms of the risk is going to be smaller. And so what we do is that we use that score to evaluate the sort of a local risk. So it's the risk between your candidate and one other sequence somewhere else in the genome that has at most 4 mismatches. Okay, so for all the sequences that have at most 4 mismatches compared to your candidate, you get that local score. And then in the second step, you combine all these local scores, and that gives you, using that formula, and that gives you the global score for your candidate. That score is between 0 and 1, and the higher it is, the less likely you are to have off-target modification. In that paper, they put their threshold around 0.6, saying that if the score is higher than 0.6, the target should be fine. But as I said at the beginning, we took the approach of being very strict about our filtering steps, and so we decided to use a score of 0.75 as the threshold. So if the target has a score below 0.75, we said there might be some risk of off-target modifications, and so we reject the target. That process is the most expensive part, computationally speaking, because we have to evaluate, to compare our candidate, with all the other possible sites in the genome. And in a mammalian genome, you would have hundreds of millions of possible sites, and so that's very slow. But the way we improved it is that we don't really care about the exact score if it is going to be below 0.75, and so we have an early cutoff point. When we calculate that score, as soon as it drops below 0.75, we reject the target, and we don't try to actually calculate the exact score. And by doing that, we save quite a bit of time. Once we've done that, all the candidates that have made it this far into the selection process are what we consider suitable targets. We check that we're happy they're going to be efficient. We check that the off-target risk is low enough. So it's targets we're happy to use. So what we do is we save these targets along with their exact location, all the properties we assess. So we save the secondary structure, we save the off-target score, and so on. We save all that into a file, and then that file is used to build a database that contains all the targets from the entire genome. So we've released that for the mouse genome, and so it's available at CRISPR.RIKEN.jp. And so in terms of stats, what we found is that for most genes, we found a lot of targets. So some genes we've found, even with these strict rules, we found hundreds of targets. But the interesting thing is that for roughly 72% of the genes, we found at least 6 targets. And so in the context of triple targeting strategy, that's something that's quite useful, because that means you can reproduce your results. You can use 3 targets to generate your knockout. And then if you find an interesting phenotype, well, you can generate a new knockout using a different set of 3 targets, and then check if you observe the same phenotype. And by doing that, you're reducing the risk that the phenotype that you observe is because of an off-target modification, because it's very unlikely that the independent set of targets would have the same off-target modification, because the targets are different. So having, for most of the genes, that ability to have multiple sets of triple targets is very valuable. And overall, we have more than 80% of the genes for which we have at least 3, so we can use the triple targeting. And then for we have under 20% where we have less than, between 0 and 2 targets. And so within that, most genes have at least one target. So that means we cannot use the triple targeting, but we can still target them using traditional approaches. So we use that strategy a lot. So I'm just going to go through some results we've had with knockout mice in the sleep context. So the first thing that we checked is that our tyrosinase knockout, so if you remember, it's the one where we just changed the color of the fur, have normal sleep, and they do. They have similar behavior to wild type, which is reassuring. It means we didn't, changing the color doesn't affect their sleep, so, which is as we expected. Then we looked at traditional clock knockouts. So we looked at BMAL knockouts, CRY1, CRY2, and PER1, PER2 double knockouts. And we observed the similar results to what had been reported in the literature using traditional knockouts methods. Then we looked at the hypocretin knockout mice, and they have a fragmented sleep, which is what we expected, because that's the gene responsible for narcolepsy. And so with all these different knockout lines that were generated, we were confident that we could use that triple targeting strategy to have efficient first generation knockout. And so based on that strategy, that we were happy with at that stage, we looked at other genes. And so we identified NR3A as a short sleeper gene. So knockout of that gene is reducing sleep by more than an hour and a half. And what we found is that what's changed is the transition between from wake to sleep. So once the mice are asleep, they sleep normally, but it takes them longer to fall asleep. The interesting thing is that because we have multiple targets thanks to our detection method, we were able to reproduce the results with a different set of 3 targets. Still, to make sure we looked at off-target modifications, we couldn't find any. And then we did mass spec analysis and confirmed that the protein was absent. So we were able to really be sure that the phenotype that we were observing was because of that specific knockout. And then we took it further, still in the sleep context, and we look at sort of different genes for different channels and pumps related to calcium channels. And so for each of these, same thing, we were able to use a first set of 3 targets, generate a knockout, detect the interesting phenotype in the first generation, use a different set of 3 targets, first generation knockout, and confirm the same phenotype. So very quickly, in the space of a few years, we looked at maybe more than 30 different knockouts, where previously each individual knockout would have taken month and month. So it's really completely changed the way the lab was able to operate. So if you want more details, all these different knockouts were reported in these 2 articles in Cell Report and Neuron a couple of years ago. Of course, when we started developing our method, the reason why we had to develop our method was that there wasn't so many tools that we could use. But since then, that has changed. Many people got interested into the bioinformatics side of CRISPR. And so we recently started comparing our method against other methods that are now available. And what we found is that our method is relatively fast. And I'll get back to that in a second, but there are some tools that are still faster than what we're doing, and so we want to sort of try to see how we can improve our method. And what we also found is that we report fewer targets than many other tools. On average, maybe 1/5 of what many other tools would report. But we don't really see that as a disadvantage of our method. In anything, it's probably a good thing, and it's a consequence of having very strict filters. Many targets that other tools would report, we reject, and we think that we rejected them for a good reason, especially, for instance, if it's around off-target risk, we think it's better to be strict and reject them. And also because the scoring method that we use for the off-target risk has been experimentally validated. It's based on that Nature Biotechnology paper from 2013. We're quite sure that we're evaluating the risk correctly, and so we think it's probably a good idea to, if we think there's an off-target risk, to just not show that target at all, instead of the approach taken by some tools, which is to report all the targets and just tell you, well, this target has X number of sites with 1 mismatch, Y number of sites with 2 mismatches, and so on. Well, it might be difficult for the user of this tool to interpret whether that's a bad thing or not. What's the limit? What's the threshold? Should I use that target, or is it going to be risky? So we think it's better to really evaluate a priori the targets and just not show at all the ones that we think are going to be at risk of having off-target modifications. In terms of things we're still working on, as I said, the method is quite expensive. So in terms of computational cost, at the moment, it takes roughly 12 days to process a whole mammalian genome. if you're doing a one-off analysis of a reference genome, that's probably fine. I mean, for the mouse genome, well, it took us 12 days, but now we don't have to do it ever again. We have all the targets for the MM10 genome. So that's fine for reference mammalian genomes. But some reference genomes can be quite large, especially for flowering plants. And so our current methods for these genomes would still take weeks or maybe months. So that's obviously not ideal. But there are also some applications, sort of long-term applications, for instance, in precision medicine, where we would work with non-reference genomes. And so we need to, and if you're working with multiple genomes like this, you want to analyze each of them, well, you probably don't want to wait 10 or 12 days every time. And so much of our current work is focused on making our method faster. And so our objective is to basically bring that, our current method to less than a day to process a mammalian genome. That's sort of our benchmark. And we think it's going to be completely feasible. The other things we're working on is the target scoring. And that's because sure, we designed the method to filter out targets. So all the ones that are left we're fairly happy with, but it might still be useful to rank the ones that we keep. It's not because they're all going to be efficient. We think that they're all going to be equally efficient. There might still be things that differentiate them. So there are some tools that have some efficiency scoring, for instance, looking at machine learning. And so we're trying to see how we can incorporate elements of that into our method. And the other thing is that we're moving past knockouts, because in our method, the only thing that's specific to knockout is the triple targeting. But all the actual selection steps for the target design can be generalized. And so, for instance, we've worked on producing a map of over the whole genome, the whole human genome. We produce a map of targets that are allele specific for the K562 encodes cell line. And so the idea here is that we have the same filtering steps. The only thing that's different is step one. Instead of looking at exons that are shared between isoforms of a gene we want to knock out, we're looking at all the allele-specific sites across the whole genome, and then we only look at targets in these regions. But the rest of the method can be easily generalized. So in summary, well, CRISPR, because it's easy to use and it offers, in theory, high efficiency, it has completely changed the way knockout lines are produced in mouse research. And of course, it changed editing as a whole. And so it's really changed how people in the circadian or sleep fields are working. And what we've shown is that by using specific strategies such as the triple targeting, we can have a very, very high knockout efficiency, even in the first generation. But what that means is that we need to have access to several targets for potentially a very large number of genes if you're looking at complex pathways. And so we developed an automated method to detect all these targets. And as I mentioned, the advantage of that automated method is that we can also increase the reproducibility of the work, because it's easy to just reproduce the results with different sets of targets because you have all of them available whenever you want. And now that that method has been validated and has been giving good results, we're focusing on improving that method and applying it to other domains. And with that, I just want to thank you all for your attention and give a big thanks as well to all my collaborators on the experimental side who made all that work possible. Thanks.
And thank you, Dr. Perrin, for that informative presentation. We will now start the Q&A portion of our webinar, and we'll address some most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question drop-down box located on the far left of your presentation window Type your questions into the box that appear on the screen and click that send button. Our speakers will follow up with you with your questions via email. So let's take a look at our viewers' questions. Dr. Perrin, what if a gene has few or no targets that pass all filtering steps?
So, I mean, our initial approach is to be quite strict. And we found that, as I mentioned, for more than 80% of the genes, even with these very strict rules, we can generate enough targets to be able to use our triple targeting strategy. But the idea is that, well, it's nice to be strict initially, but many of these rules can be made more flexible. So if there is a specific gene that we want to target, and with these very strict rules, we don't find anything, then we can just rerun the method just for that gene with some relaxed rules. And we know that there's a risk in doing that, but it's we only do it for the genes for which we don't have enough targets. So we have the ability to on-demand rerun the method for just specific genes with relaxed rules, just so that we find something.
Thank you. And Dr. Perrin, how can speed be improved?
So at the moment, so in the 12 days, the two slowest steps are the last one. So the secondary structure analysis and especially the off-target scoring is really the slowest one. And as I was mentioning, it's because for each candidate that we're evaluating, in theory, we have to compare it with all the possible sites in the whole genome and then check for each of them, do we have less than 4 mismatches? And if yes, then we have to score it. And because in a mammalian genome, we have, for instance, in the mouse genome, we have 245 million such sites. Well, for each candidate, we have to do that 245 million times, potentially. And so that's what takes a long time. But what we're working on is basically clustering strategies to, for one candidate site, only consider the sites that we know will have less than 4 mismatches. So some sort of pre-processing steps. So that's, instead of scoring one guide against 245 million sites, we only score it against the sites that we know are going to be a risk. So basically focusing on pre-processing methods to speed up that step. And because we spent most of the time in that step, any improvement that we make is going to drastically reduce the overall time. So we think it's quite realistic to aim for processing the whole genome in less than a day. We're also focusing on optimizing the code. So part of it is developing in Python, which is was easy to develop for initial development, but it's not necessarily the most efficient programming language. And so we're porting part of that code to C, which is also going to give a performance boost.
Thank you for that. And Dr. Perrin, can the method be applied to any organism?
Yes. So most of the initial work that we've done was on the mouse genome, because that's what the group was using. But there's nothing in the method that's specific to the mouse genome, so it can be used with any organism. So since then, we've worked on human cells. So for instance, K562 is a human cell line. We're also working with bacteria, with plants. And so there's nothing specific to the mouse in our method. Basically, any organism for which you have the genome as an input, we can run the method on it.
Dr. Perrin, thank you so much for your presentation today. Would you like to provide our viewers with a closing remark?
Yeah, I mean, as I said, for the genomes that we've already analyzed, so for instance, for the MM10 genome, there is a publicly available database where you can just query any gene that you're interested in. But what we're also working on is a release of the code, so that if you have one genome that you're particularly interested in that is not available online, you can just download the code and run it directly on your genome. So that's going to come up shortly. So if we if you're interested, you can just send me an email and I'll give you the link once it's released.
Dr. Perrin, thank you again for your presentation today and for your important research. I'd also like to thank LabRoots for making today's educational webcast possible. Before I go, I want to remind everyone that today's webcast will be available on demand viewing through January 2019. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share this announcement with your colleagues who may have missed today's event. That's all for now, and thanks for joining us. We hope to see you again soon. Bye-bye.